Shalom Maya and Ronen and all the students joining us. You ask me how biological databases can be used in order to gain medical insights. The example of molecular mimicry is indeed interesting. This process may be initiated when a foreign protein, such as protein from bacteria, activates the immune system. If by chance this protein shares a sequence or structure motif known as epitope with a human protein, then the activated immune system will continue to attack the similar human protein even after the original infection is long gone, leading to autoimmune episode or even long-term autoimmune disease. Actually, several years ago, I was involved, together with Professor Steve Melnick from the Kaplan Medical Center, in a study where we suggested molecular mimicry as a possible mechanism for perimyocarditis following streptococcal A infection. The extent to which molecular mimicry contributes to autoimmune disease is still being debated, but I would like to discuss a technical aspect of this study that relates to bioinformatics. How did we find the bacterial protein that is similar enough to human cardiac protein to invoke such a reaction? This question introduced the larger issue of sequence and structure databases and how we can search them. So, in this unit, we will talk about biological databases. In the first segment, I will introduce databases that store information about biological sequences and molecules, mainly GenBank for DNA sequences, UniProt for protein sequences, and PDB for protein structures. As you will see, the sequence databases are so large that we need special computational tools to search within these databases, and thus the second segment will discuss BLAST, which is an outstanding algorithmic tool that enables us to perform such searches at amazing speed. In the third segment, we will describe several additional databases that are relevant to human genetic variation and human disease, and describe genomic browsers, which incorporate many layers of information, including information about such human variations that are built on top of the primary sequence. Even in the early days of molecular biology, people understood the importance of databases that would store the acquired information. However, no one could envision the growth of such databases and their increasing complexity. In the beginning, a large number of such databases were started as private initiative by individual research group. A research group that specializes in a certain type of protein or certain group of organisms would to collect relevant sequences. However, such initiatives were often abandoned when the relevant PhD student graduated or when the lab ran out of money or changed focus. Thus, it became clear that there is a need for central hubs that would maintain biological sequences, set the standard of submission, maintain older versions for compatibility, and manage these databases professionally using state-of-the-art techniques. In 1988, in parallel to the inception of the Human Genome Project that produced sequences data in unprecedented amounts, the NCBI, the National Center for Biotechnology Information, was formed as part of the United States National Library of Medicine. GenBank, which is a depository of DNA sequences that started as a local initiative in Los Alamos in 1982, was transferred to NCBI in the early 90s. Another major incentive for the growth of sequence databases was a decision around 1989 by the leading scientific journals and funding agencies to require the position of all discovered sequences in a public database as a condition for publication. Since scientists are motivated by grants and publications, this step contributed significantly to the creation of bioinformatics community in which genomic data is freely accessible. In 1987, the entire content of GenBank was printed in a set of 7 volumes. The rapid growth in the size of GenBank is such that today GenBank contains more than 279 billion bases that form more than 200 million genes from tens of thousands of organisms. This amount of information would fill roughly 120,000 book volumes. It is therefore no wonder that no one is printing the contents of GenBank anymore. Each GenBank entry contains not only the sequence itself, but also information about its genomic location, related PubMed publications, information regarding the exonic structure of the gene, and more. NCBI maintains a suite of related databases. For example, the assembled genome of thousands of organisms, including, of course, the reference version of the human genome, a list of more fully annotated genes called RefSeq, and several more clinically related databases such as OMIM, dbSNP, and ClinVar, which we will discuss in a later segment. While the NCBI also maintains a database of protein sequences, many scientists prefer to access protein sequences via UniProt, which is maintained by the EBI, the European Bioinformatics Institute. The data are comprised of two collections, SwissProt, which currently includes about 500,000 sequences of proteins that have been manually annotated and reviewed, and thus their existence is confirmed, and Tremble, which currently includes about 137 million protein sequences that have been computationally translated based on nucleotide sequences. While most of these Tremble entries likely represent real proteins, their existence has not been validated. The figure shows the striking growth of Tremble over time. The temporary decline in the number of sequences in 2015 was a result of a cleanup of redundant entries. While we are usually interested in the sequence detail of specific proteins, the database can also give us additional insights. For example, the distribution of protein lengths shows that the most typical size of a protein is between 200 and 300 amino acids, an observation which have evolutionary implications. While the content of sequence databases is straightforward, containing the sequence of actual nucleotides or amino acids, structural databases are more complicated. They have to store the three-dimensional coordinates of all atoms of the DNA or protein molecule, the coordinates of ligands when present, and additional experimental details describing the X-ray or NMR experiment used to determine the structure. PDB, Protein Database, is the main database that maintains these structures. As you can see from the figure, it currently has about 150,000 entries. However, many proteins have been solved several times in complex with different ligands or from highly related organisms. Thus, the actual number of unique proteins for which we have structures is between 30,000 to 50,000. The range of numbers reflects the range of exact cutoff used to define which proteins are considered different. In the early days, the database consisted simply of files containing a list of the three-dimensional coordinates of each atom in the protein. Researchers could download this list and visualize the structure using molecular display software. Currently, the PDB has become a much richer source of information and includes a built-in simple molecular visualization tool that allows users to directly view the structures. Today, all these databases are cross-referenced, and from a gene entry in GenBank, one can move to see the corresponding protein sequence at UniProt, and then follow a link to explore the structure, if it exists, in PDB. Now that we know more about the organization and content of these databases, we need to understand how we can search efficiently for the information we need in such huge databases. This will be the topic of our next segment.